despite I have a primary talk, I actually decided to sneak in some ongoing projects. So we'll start with brief introduction and then go to some study that we're doing right now. So what is mutation? Mutation is difference in the DNA between maternal cell and daughter cell, and like division starts, DNA duplicates, then it's split into cells. And if there is difference in DNA between this cell and ancestral cell, this is a mutation. And surprisingly, cell division, DNA doubling, is extremely precise process. And at least if we're talking about germline, if we're talking about cells that became gametes, mutation rate per division is, per base pair per division is extremely, extremely low. And this high precision actually allow us not to like have the same DNA between different cells in a body. So like if precision of replication would be much lower, we will have very, very high mutation rate between different cells in our body, and that will cause a problem. Also, like high mutation rate between generation will lead to tons of genetic disease in offsprings. So there is probably a lot of evolutionary pressure to have such a low number here. Yeah, so mutations can be subdivided on two categories. One category is germline mutations that can be passed from the generation to generation. This mutation occurs in sperm, and if mutated sperm or oocyte, if mutated sperm or oocyte actually became a new individual, then like this mutation goes to population. And in alternative to germline mutations, there are somatic mutations that occur in cell in a body, can be inherited, but may affect like substantial amount of cells if it's happens early in development. And germline mutations are very important for evolutionary biology, population genetics, because like this mutation actually come to population and is source of genetic disease. And the most important reason why we care about somatic mutations is cancer genomic. I believe that there's like a lot of different applications, but like the most famous one is somatic mutations fuels abnormal cell development in the body, which actually may lead to cancer. I believe there is conceptually 3 big different reasons why we may think that mutations is worth studying. First of all, we need good statistical model for mutation rate to discover genes driving disease and evolution. Second reason is deep understanding of mutagenesis, of underlying biology, can help us to design therapeutic and lifestyle interventions. And last reason that actually mutational patterns may be used as readout of cell properties. So let's go. Okay, let's go through these points. So in 2012, gene CSMD3 was associated with lung cancer. And this happens because there was excessive mutations in this gene, like compared to the, let's say, flat estimation of mutation distribution across the genome. However, mutation rate distribution actually extremely variable across the genome. And when Gadi and Shamil and other colleagues built better mutational model, it became obvious that mutation rate in red, that this gene just have very high mutation rate, and excessive mutation in CSMD3 can be just explained by high mutation rate in the gene. So better understanding of underlying mutation model help us to discover disease-associated genes. The same problem actually happens in germline mutations, in germline mutagenesis. When people trying to find genes associated with autism, we need precise mutational model to actually distinguish genes with high mutation rate versus genes that happen to have a lot of mutation by chance. Yeah?
So when you talk about the mutation rate, is this sort of without selection, or is this observed mutation frequency after cells have been growing in the population?
So I'm talking about mutation rate as underlying neutral process, so before selection. And that's why mutation model is very important, because if you saw a lot of mutation in a gene, you think, okay, this mutation happens because of the selection, because like there is a cancer cell and it was positively selected, and that's why there is a lot of mutation in this gene. But actually, like if a lot of mutation may happen in this gene by chance, like you may be confused unless you have good mutational model. If you expect a lot of mutation by chance, you can't distinguish between selection adding mutation to this gene versus just neutral mutational model. Okay?
Or on the other extreme, just to understand, if suppose that a mutation was lethal for a cell.
Yeah.
You wouldn't say the rate was 0. You would just say it was whatever it was, even though you wouldn't observe any cells with that mutation.
Exactly. It's definitely not easy to measure, but...
Sure, but just an extreme example to understand that.
Yeah, exactly. So we would say that this region have non-zero mutation rate while we don't observe any mutation because of negative selection.
Well, you would observe synonymous mutations there. So you'd say like something like DNDS would be less than 1.
I was thinking of just like one really specific, like, let's say loss of function. But sure, next door neighbor, synonymous.
Or just across.
Or right there. Yeah.
The gene as a whole is under negative selection.
Yeah, like definitely, like you can use, and DNDS actually assume that you normalize by neutral mutation rate, right? Like you compare number of mutations inside in positions that affect gene function versus positions that don't affect gene function. So you basically normalize on mutation rate that you consider to be neutral. However, it's not works perfectly, to be honest, because the context distribution around synonymous position, non-synonymous is relatively different, and you still need to build precise mutation model to account for this. And there's also like other problems with just sampling noise. And if you have just better regional mutational model, that's help to overcome this problem. Okay, so first one, it's applicable to discovery genes associated with disease. Second, it's helped to find biologically relevant process that may lead to some disease. So this is a recent paper from Gadi's lab when somatic mutation from the skin was called with tricky algorithm. And there is difference in mutation rate in sun-exposed skin versus non-sun-exposed skin. And it clearly says us that UV light, the exposure to sun, is mutagenic. And after that, we know that how, for example, melanoma occurs, why it's related to the time that you spend on time, under the light. Another relatively different or relatively recent discovery is about detrimental effect of traditional Chinese medicine. There is like one specific traditional Chinese, there is about aristolochic acid. There is like component of some herb medicine, and it's just strongly increased mutation rate, at least in liver, and that's why it's caused the cancer. And after we know this, we can't prevent people to take this traditional medicine because we know that it's mutagenic. That's why it's caused cancer. And last one, you can use mutational pattern to understand underlying cell biology. There is very different cell types in our body. And different cell types actually have different mutation rate distribution across the genome for whatever reason. And if you know the expected distribution, you having just mutation from the tumor cell, you can predict from which organ this tumor cell comes. So if you have metastatic cell that you don't know to which, from which organ it originates, you can backtrack the cell of origin of this metastatic cell, which is like actually a relevant thing for the to treat cancer. Okay, so that's why mutation importance. Then let's talk how they occur. And there is two major processes that contribute to them. First of all is DNA replication, DNA doubling. And DNA doubling realized through the antiparallel DNA synthesis. It's semi-conservative, so you have original DNA like put it as a bubble, and then new DNA strands synthesized based on original DNA molecule. And this process definitely may introduce mutation by itself because the proteins that double in DNA have some error rate, and like this is one source of mutation. Another source of mutation is just DNA damage. So DNA in healthy cell in normal condition accumulate about 1 million lesions per day, and definitely these lesions can be converted to mutations. However, and there is a lot of very specific defenders against these lesions. These safeguards called repair systems, and they're extremely effective in removing DNA damages. So just to add some context here, there is like few hundreds of proteins, different proteins that participate in DNA repair, and if you mutate few of them, the mutation rate bumps. For example, here are the cancer samples with mutation in one gene. This is cancer types with mutation in another repair gene. And that's how mutation rate increases in these two cases. So like it may increase by a factor of 10 or even 100. So there is actually two different categories of DNA damages, and one of them is point lesions when one nucleotide affected, and another one is when DNA breaks. And there is a ton of repair system, and actually with each of these repair systems there is many sub-pathways. So it's really complicated process with a lot of redundancy, and it's actually really hard to say which contribution of which repair system, what contribution of which repair system in healthy cell, for example. So I will talk a little bit about two systems to tolerate DNA lesions. First of them is translesion synthesis. So if there is DNA lesion coming to replication, it's caused DNA replication obstacles. And to overcome this, not to die, cell need to do something with this lesion. And even if replication keep going, introducing mutations is much better than just cell die because it cannot deal with this lesion. And the system to tolerate the lesions, one of the very active, is translation synthesis. So when the normal replicative polymerase meet lesion, it stalls, and then it's switched with the polymerase with very low fidelity, but this polymerase actually can bypass this lesion. And what happens is introduce mutation opposite with high probability, is introduce mutation opposite initial lesion, and it also may introduce additional mutation because it's low fidelity. However, it's much better for the cell to tolerate lesion in this way than just, again, cannot continue replication. Another system is nucleotide excision repair. This is like complicated cascade, but
I have a question on the previous slide. That is active, can that be active both on the leading and lagging strands?
Yeah, it's active both on the leading and lagging strand, and actually we believe it's more active on the lagging strand, slightly more active.
So I suspect if you stay for the second talk today, this will be mentioned. And Vova has a paper in Nature Genetics earlier this year exactly about this effect, what you're asking.
Okay.
That's exactly correct observation.
Okay.
Does it go back to the old one? Like, how long does it take before the polymerase goes back to the usual one?
We don't exactly know, but usually not much longer than hundreds of nucleotides. Okay, I should speed up a little bit. So another system is nucleotide excision repair, and there is, what's important, there's two subpathways. And subpathways is different in a way how lesion detected. First branch is global. Okay, second branch is left one. When lesion detected with some systems that working all over the genome, and this called global genome repair. And another system when lesion detected with transcription, with RNA polymerase during transcription. So RNA polymerase stalls on the lesion, and there is a lot of proteins recruited to perform repair. Yeah, okay, I don't have time, but I will do this. So there is tons of proteins here that probably not, like their name don't say you much, but actually these two proteins called CS, A, CSB is proteins that actually called Kakayana syndrome A and Kakayana syndrome B, and this is very severe neurodegenerative phenotype. And tons of protein called XPA, XPB, XPC, XPG. This is different subtypes of xenodermal pigmentosum. So like at least 9 proteins out of the system manifest different phenotypic syndromes that are very severe. And why I'm talking about this biochemistry, because the knowledge about it can be used to understand how to start with real data and project it to biochemistry. So transcription coupled nucleotide excision repair, very effective on the transcribed strand of the gene and non-effective on non-transcribed strand of the gene. And what happens in real data when there is a lesion that can be repaired by this system, for example, lesion introduced by tobacco smoking, the rate of mutations that caused by tobacco smoking is very different on transcribed and non-transcribed strand of the gene. And usually this difference means that returning back, usually the difference between mutation rate on 2 DNA strands that one of them replicated and another one not, is usually mean that these mutations associated with bulky lesions.
I have a question.
Sure.
Do you know, so a lot of enhancer and regulatory regions, active ones at least, are transcribed as well. Do you know if the mechanism is also active there, or is it?
It's also active. So, for example, at least like with my hands, I'll look on the promoters at least. And in promoter, there is synthesis in opposite direction. And actually, how asymmetry looks, it's in one direction in the gene, and it swap in opposite direction if you pass transcription start site. So that's actually the case. And if there is unidirectional transcription enhancer, I'm pretty confident that it will work exactly the same way.
So I can add that promoters are regulatory sequences. There is also a possibility, which you all probably will mention on the next slide, that due to chromatin accessibility, there is facilitated access of the global system. So not only transcription coupled system, but the global system can be more active as well. And sometimes tight protein binding is possibly blocking the access of anywhere. So in genes it's primarily transcription coupled, and in regulatory regions it's both transcription coupled and maybe increased activity of the global system.
Okay, and basically last few last introductory slides. If you also can use different footprints of DNA repair, if someone experimentally measured efficiency of DNA repair, like in this case, this slide's in blue, then if there is mutation rate that's anti-correlated with the efficiency of DNA repair, you can probably say that this mutation is actually caused by decreasing the efficiency of repair exactly in this region. And that's what was discovered pretty recently in cancers. So I talk a lot in introduction about discoveries in cancer, but actually our goal trying to do this in the germline because, again, understanding biology of the germline mutations is even, I believe, even more important, and we basically know nothing about contribution of different repair mechanisms and systems to the mutation rate in the germline. So our goal actually, like both my talk, the rest of my talk and Ruslan talks would be exactly about this. We will try to find contribution of different mechanisms to the patterns of mutation rate or mutation type distribution across the genome. Okay, so let's talk a little bit about the research. And I will talk about clustered mutations. So what is clustered mutations? Actually, if DNA doubles, mutation occurs not just independently, but rarely few mutations occurs very close to each other. And saying rarely, they occur much more frequent than you will expect by chance. And we believe that this actually sign of non-independence of mutations, and it means that they occur simultaneously during one biological event. So back in the past, Shamil did this very simple and very efficient test. They compare distance distribution between mutations within the one offspring and distance distribution between mutations between non-related kids. And
So these are de novo germline mutations?
yeah, this is de novo germline mutation. And what was found that if you look on mutation within one individual, the frequency of mutations at very short distance much higher than if you compare mutations from non-related individuals. And this is probably a sign of non-dependence of mutations, not because of the variance in mutation rate, but because of some drastic events that caused many mutations simultaneously. Also, recently there was a paper, actually I participated in this one, when we discovered that if you look on clustered mutation rate, it's extremely heterogeneous. That actually data about de novo mutation rate, so mutations that occur in a kid compared to the parents. And this is clusters that coming from oocyte from mom. This is cluster coming from the father. And as you can see there, the distribution of mutation of maternal clustered mutation rate extremely not flat. There is huge bump right here. And we hypothesize, probably incorrectly, that that this mechanism related to double-strand breaks. So we said that probably this region is susceptible to double-strand breaks in oocytes, and that not so frequently happens in sperm. Well, I will.
Is that like one part of one chromosome? What
Yeah, this is chromosome 2 and scale about like 100 megabase. Yeah, I definitely.
Yeah, Alex, it's not the only region of the genome, but they all show the same spikes. So it's very localized process, but it's not the only place in the genome. They are spread out.
Okay.
Is there a nucleotide context bias to these clustered mutations?
Yeah, there's a lot of biases in mutation type distribution and context biases in cluster. But we still don't know what drives this bias.
I'm just thinking, if you can assign them to a process that's known to specifically target single-stranded DNA, like APOBEC, which has a very characteristic trinucleotide context bias.
It's not APOBEC.
Even in the literature, it's APOBEC, it's not APOBEC.
Well, I'm not saying it is, I'm just giving that as an example of a
You're not the
process.
only-
Yeah, right. So I think what happens is the rest
We
of the
put some
problem is
effort.
exactly what you're suggesting.
So we put some effort to say that it's one protein and another, and we not succeed. And we instead succeed with another hypothesis that end up to be wrong, so like be accurate. So how I try to look on cluster mutation in this study, there is few mutations that close to each other. And I want to consider them as just sequence of mutations, ignoring the position of the genome, at least for starters, or distance distribution between mutations. It's just like this cluster should be written as AC, GC, TG, and so on. So like just sequence of 12 types of mutation in each position. So how we extracted this data, we use human polymorphism and look on the scenarios when two mutations occur close to each other, a distance up to 5 kilobases, and present only in 2 or 3 individuals out of hundreds of thousands. So that's like working definition of what we consider cluster in this specific
So
study.
you use rareness as a substitute for de novo, basically?
Exactly. Yeah, that's the whole idea. And the short special distance to enrich it with actual cluster mutation, because you can consider like mutations that far from each other, they still would be on one haplotype if they're rare, but like because cluster mutations dominate short distance and became like less frequent on longer distance.
And it's top-med phased?
It's top-med phased. Yeah.
Top-med is phased?
Top-med is phased, yeah.
Okay.
It's-
These are rare enough that it would be hard to impute on them. But I assume that since you
No,
only...
this is these are 100,000 whole genome sequences.
But it
They're not,
depends.
it's not, you don't need to impute, it's sequence.
And actually, that's not very important, because if you think that there is two specific individuals, you can assume that there actually happens on the same haplotype of these two specific individuals, even without phasing.
Not
You
predictive.
even phasing or imputation? You even phase or impute?
I mean,
You don't have to
impute phasing. Because obviously you're not going to have a paradigm grid that physically
Yeah,
phases things.
that's correct. You generally would not.
Right.
But they're rare enough that since you only see them together, you can assume that they are indeed in sync.
Exactly. So actually, without phasing, this assumption still will work, that it usually will occur in the same haplotype.
So numerically, at least in the novel data in the paper oversight, it's 2% of mutations come in clusters. So it's amazing enrichment. So if you're looking for such events, right? So because your expectation of a cluster is so high, that most of those would end up as clusters.
Yeah.
Because if clusters were very rare, this strategy wouldn't work. You would frequently have phasing errors or haplotypes of independent mutations just being on the same haplotype. But because there are so many, we know that most of the signal we're seeing comes from clusters.
Yeah, I think those are points.
Yep.
Oh, okay, so what we're actually doing with this data. First of all, my goal was to extract some structure within these millions of extracted clusters. So I decided to fix the number of mutations in the cluster, convert clusters to the 0-1 vectors when each mutation in each position can be written in 0 and 1s. So like there's 12 mutation type, and in each position you have exactly one non-zero sign across 12 in 12 number block in a vector. So you start to convert them to 0, 1 vectors. Then you can calculate correlation matrix between mutations across all positions. So you have 48 on 48 correlation matrix, and then you do dimensionality reduction. So you project, we have 50,000 clusters that are 4 mutation long. Then we project these clusters to the new low dimensional, and in this case, low means 8 dimensional space. And we can measure the distance between clusters in this space.
Question.
Can you explain one more time the 12 by 4? What's
Yeah, exactly. So, do I have a marker? I do, thank you. So, for each mutation type, so there's 12 mutation types, okay? Single nucleotide mutation type. And you want to write this mutation type as 1 in out of 12 signs, which each 0 means that there is not other type of 12 type mutations. And then you have 4 positions in a cluster. You have clusters that have exactly 4 mutations. So you fix this. And then like you have this mutation, and this mutation would be another block in this vector. And you know, like this is C2T, that means it's like first 1 and other zeros. And then you have vector with exactly 4 non-zeros.
you chose 4?
Yeah, because this trade-off between amount of data, longer cluster, like if you consider longer clusters, that would be like much, the data just decline with the length of cluster, amount of the samples. At the same time, like decreasing the length of the cluster decreases the dimensionality of your space. So that's trade-off between dimensionality of the space and actually like sample size that allow you to extract the structure.
You probably played with 4.
I played a lot with different numbers, and actually the result that I will show, like just I will say, it's a head, they are very consistent. So you can extract the same structure from the cluster with a length of 3, and probably with length of 5, but like I believe that cluster would be
And
very.
it was just SVD,
That was SVD.
the dimension.
Yeah, dimensionality reduction was done by SVD.
So the question Camille was supposed to ask, why 12 and not 6? And the reason is because this is a strand-coordinated process, we don't collapse complementary mutations, right? So you don't even know whether it's A to G or C to T, but we cannot collapse them because we think that the cluster happens on the same strand.
Well, the whole thing could have been reverse complemented.
The whole thing.
Yeah, the whole thing. But like definitely vector couldn't be collapsed to the blocks of 6 because if you should distinguish between C to T versus C to T, G to A. This is different types of cluster.
Yeah. But the whole idea of this project in the next talk is that we don't collapse reverse complements for obvious reason for individual mutations. But this higher level symmetry, what you're implying, can be used to validate that the results make sense. So this is a very important property when you get the answer if it's not symmetric with respect to complementarity operation for the whole thing, right? It's not the right result.
Alex, how much time do I have? This is 9:45 right now. 5 minutes?
6 or 7.
Okay, generous. We extracted, so we apply clusterization procedure. We extract 7 classes, and 3 of them produce reverse complementary results. And this is just aggregated profile across 4 positions within the cluster, within the three pairs of the processes that we extracted. And the amazing thing here, that first position slightly different in the spectra compared to the rest positions. And surprisingly, this is this is process 1, and these mutations that enriched in first position compared to the other positions. This is process 1, process 2 have the same property, and process 3 we couldn't talk about this in 5 minutes time frame. And surprisingly, screening through the literature, difference in mutational patterns between first position and rest of the positions have only one reference, like to what mechanism it could be. And this mechanism specifically can be only translesion synthesis, according to the literature. Okay? There is only one known mechanism that fit difference in mutation rate distribution between positions in the cluster. And surprisingly, doing this simple procedure, we can drive the conclusion that major cluster mutational mechanism is translesion synthesis. So we basically have very, very strong prior that this is correct hypothesis how cluster mutations occurs in human germline. Is the logic correct? Is it logic understandable? Is it clear?
Because most of them have this unequal.
Yeah. So this tiny bit of imbalance is actually very significant and strong enough to make the story from it.
Maybe I can relate that. So you're thinking about possibilities, right? So one possibility is that there is a process that has very high mutation rate and very highly localized, and produces what you're seeing is a bunch of mutations produced by one process. So the other model, which we argued with Vova, is that it's a 2-step procedure. So you have first mutation, first event, that triggers cluster formation, and all other mutations are brought by a single mechanism, right? So there is a fundamental asymmetry between the first event and the other events. And if you're thinking about all possible pieces of biology, the story of recombination rate, the story of localized damage, anything is just highly localized mutation rate. What we believe is happening is not highly localized mutation rate. There is a first event that triggers cluster formation. Now, the only event what we know from biology is that if there is damage on DNA, which triggers translesion synthesis, this is the event that switches to low fidelity polymerase that has high mutation rate. Now, then there are 2 possibilities. One possibility is that there is a damage that is being then repaired correctly, so you don't see the first event. So in this case, first event would be similar to the others. And the other possibility is that the first lesion also leads to mutation. So this admixture of the second class, when the first lesion leads to mutation, generates this possibility that you have one spectrum for mutation that is number 1 along DNA sequence, and all the others are identical to each other. So this model only fits one process, because we believe it's 2-step, and the only known 2-step process is translation. So that's, I don't know if it's clear, but I'm trying to kind of clarify the logic here.
Yeah, and I will show just last piece of this work, actually most recent one, that you surprisingly can measure mutation rate infidelity of this polymerase that introduced these mutations. How you can do this? If you consider, the start of the cluster, because it starts with very specific mutation that's associated with deletions have very different spectra to the rest of the mutation in cluster. And then at specific distance from this cluster, you can calculate, just count number of occurrences when exactly one mutation happens in some region, some fixed region, and number of occurrences when 2 mutations happen in some region. Ratio between these two scenarios gives you exactly mutation rate within the region. So you actually can measure properties of the protein that generate these mutations just from population data. Like you cannot measure, for example, point mutation rate from the polymorphism data, but you can measure mutation rate by the translation polymerase. And that the measurements, like the plot, how mutation rate decline with the distance from the start of the cluster. And actually this estimation that we obtained bioinformatically perfectly in line with the experimental data for polymerase zeta, which is translation polymerase, and like have a lot of measurements about its activity in experimental systems. So we somewhat recover. We probably can make bets that this polymerase is polymerase epsilon because of the mutation rate and type of mutation introduced.
What's that?
Zeta, sorry, this is zeta. Okay, yeah, thank you. I'm already out of time.
Just a quick summary from Vova's introduction. There are major two sources of mutations, either replication errors or unrepaired damages, and this is a whole complex process. There are different sorts of DNA damages that constantly pressure genome integrity, and there are a variety of DNA repair systems that preferentially tend to repair different sorts of damages, and they're generally efficient, but sometimes they also make errors. And replication is a whole difficult process. And so dozens of years of biochemical studies, mostly in model organisms, basically characterized and described properties of many biochemical pathways that generate mutations. However, despite these all efforts, we surprisingly little know about mutational mechanisms that actually active in germline cells. And but now we have a lot of sequencing data on germline mutation. We have data from studies of de novo trios, we have data on population variation, and we have data on interspecies divergence. And so the goal of this talk is to somehow, from statistical properties of this data set, to understand what are actually mutational processes that generated them. And so formally, mutational process has two characteristics. One is a spectrum. Spectrum is just a probability or propensity to generate different sorts of mutations. And in our case, we consider single nucleotide mutation and its context. It's 3' and 5' nucleotides. So we totally have 96 different types of mutations because there are 6 different types of point mutations. We collapse. Not we, just traditionally people collapse complementary mutations. And there are 4 options for 5' and 4 options for 3' nucleotides. So totally 96 different types of mutations. And here are a couple of examples of mutational spectra of different processes. This is, let's say, ultraviolet, which is dominated by C2T mutations. And you can see from here, for example, there are that C2T mutations tend to happen in a very specific nucleotide context. That's why basically context matters. Let's say there is a smoking-induced mutations which are dominated by C2A mutations. So this is one property. Another property which is important for statistical inference is exposure of DNA sample to this mutation process. Let's say one DNA sample can be, such as let's say in case of UV, skin is really exposed to UV, but let's say cells of liver or germline likely not exposed. And surprisingly, this simple mathematical framework was already sufficient to get really deep insight in cancer biology. And I'll just briefly introduce how people used it there. So we can summarize mutations that happen in a tumor sample as just a is just a vector of frequencies of different types of mutations. Here I just show, instead of 96, just 6 types of mutations for simplicity. And we assume that these mutations actually come from a small number of processes, such as each process is characterized by a unique spectrum and contribution to this sample. So that's why we expect that all this vector of frequencies is a linear combination of unknown mutational processes, of unknown spectra of mutational processes with a known contribution. So the key here is that different samples have usually a quite different contribution of mutational processes. And here what you see here. And we can use this differential exposure in different samples as a signal to extract underlying mutational processes, if this model is correct. And so the simple mathematical formalization is non-negative matrix factorization, where matrix of frequencies of mutation types per different samples is decomposed in a product of two matrices, of two low-dimensional matrices of mutational spectra of non-mutational processes and intensity of these processes in tumor samples. So it turns out that this simple framework allows us to recover spectra and intensities and reconstruct many mutation processes that people have described before in mutagenesis. And definitely it kind of led to dramatic success in this field. So the similar attempts were done for germline cells, but they were not that successful. And it seems that the reason why they were not that successful, that different individuals have quite similar patterns of germline mutations. And the reason because probably mutational processes that operate in germline cells in different individuals, they're quite similar. So it kind of really didn't work out well. But there is another property of germline mutations that can give us sufficient statistical signal. It is well known that the rate of germline mutations vary along the genome, and here is an example of mutation rate for different mutation types along the genome, and it's quite notably variable. And so we hypothesize that we can use this variability as a statistical tool to extract mutational signatures. And formally, to do so, we hypothesize that observed rates of different mutation types comes from a small number of underlying processes, where each process is uniquely characterized by its spectrum, and it has intensity that varies along the genome, so that final observed mutation rates is a sum of mutation rates that come from each of these processes. And so now, having observed this, so we don't know these underlying processes. We know we observe only outcome, and we now bin the genome in non-intersecting windows, and we consider each window as a sample. So if, let's say, in cancer genomics, we considered each tumor sample as a sample, now we consider each genomic region as a sample. And so, kind of, to have sufficient power to make statistical analysis, we basically aggregated all rare SNPs as proxy of de novo mutations from a population variation dataset, from TopNet dataset, and we got around 500 million events. And so using 30 kilobase windows, we kind of got a little bit of we got more than 5,000 mutations per window, and that gave us sufficient power to interpret each window as an independent sample. And so we had enough events in each window. And we considered 192 mutation types because we don't collapse complementary. We have 12 different single nucleotide mutations, and that is surrounded by 4 different possible combinations of nucleotides on the left and on the right. So with that approach, we first looked if it actually works, at least in simulations. For that, first we actually formalized the problem. So we assume that frequencies of mutations in each window is again a linear combination of frequencies of mutations of unknown mutational processes and intensity of these processes in this window. And so that's why this whole matrix X, which is a matrix of frequencies of mutations of different types in different windows, we assume that it is decomposed in a product of two matrices of small dimensions, of matrices of spectra of unknown processes and their intensities in windows. And so how we do it formally, we basically departed from original framework of non-negative matrix factorization, and we do PCA, ICA procedure. And basically we consider our initial matrix of frequencies. We normalize mutation rates. For each mutation type, we normalize mutation rates to 0, mean, and unit standard deviation. And then we do dimensional reduction and consider first some number of first principal components and look at kind of loadings of mutations per first principal components, and then we rotate these principal components to maximize independence between them. And then we just interpret loadings of mutation types on a given component as a mutational spectrum. And here is how it looks like. So again, different bars are loadings of different types of mutations. So positive loadings indicate that mutation rate of a given process is higher than genome-wide average, and negative loadings indicate that mutation rate in a given process is lower than genome-wide average. And actually different and bars of different shades indicate mutation rates of complementary mutations. And then we can project this predicted spectrum onto a spectrum observed in window and get expected intensity of this process. And here is actually this is real data. This is intensity of this process predicted along one of chromosomes. Yes.
Can you say something quickly about the motivation for doing this Z-score PCA versus just doing NMF before?
Yeah, so we invested quite a lot of efforts in it, and we tried a bunch of approaches, including NMF, including Dirichlet allocation models, PCA, wherever. And it turns out that actually, if we use approaches like PCA or ICA, and we decompose intensities of spectra, we actually, statistically, we get more or less identical results. In a sense, number of components number of biological mutation processes that we see. If we apply NMF, or if we apply a latent Dirichlet allocation model, then we usually either don't find anything or we find only a few processes. And we thought what could be the reason, and kind of our hypothesis that actually, in contrast to cancer genomics, spectra and intensities of mutation processes in germline, they're not sparse. In a sense that kind of, in cancer genomics, what you have usually, you have very sparse, let's say sparse intensities among samples. You have, let's say, smoking-induced mutation that happen only in a very small subset of samples. Here it seems that our mutation processes that I'll show, they actually reflect some basic functions of DNA repair polymerase machinery. That's why they're active along the genome, just their contribution slightly varies. And so we have lack of sparsity of intensities, we have lack of sparsities of spectra, because these processes basically generate all types of mutations, but with slightly different biases. And we guess this and it seems that because of this lack of sparsity, the traditional NMF or LDA approaches do not work. And we basically did simulations, and I actually looked at when I kind of decrease number of sparsity, then at some point these methods actually break and they cannot reconstruct the regions.
So if I can pitch in, so an NMF doesn't give you a unique solution in the general case, right? So it gives you a unique solution if the data are very sparse and there are sparsity conditions on NMF. So we just, these conditions are not satisfied. So this is why we just don't get anything which would be unique and stable. So the reason, I don't know, Ruslan, if you'll be talking about that, the reason PCA is not sufficient is because we don't have all eigenvalues different. Because by construction, as I hope it would be clear, you get certain processes with exactly the same eigenvalue. And you have to rotate the space afterwards. Sorry,
Yeah.
I stole from the slide.
No, it's okay. I'm not going to talk about this. Yeah, this basically was the motivation.
So you only rotate within the eigenspaces?
Yeah. It's quite a weird procedure, but it's kind of statistically it delivered the same results, but biologically it gave the most meaningful outcome. And so,
besides the lack of sparsity, is there sort of a meaning to positive and negative values in the intensity in terms of like enrichment and depletion from the genome-wide average? Do you think that's also related to why you want to sign?
you mean meaning and
It's meaningful that there's sort of positive and negative values.
it's meaningful because yes, it's again, think about this as biases. You know, a process generates all types of mutations, but it tends to generate slightly more mutation of one type and slightly less mutations of another type. That's why I think it makes sense, and it's in line with this vision that we have biases of mutation rates, not drastic differences. If any more questions? So we can move on, and we first checked if this approach works, or in a sense, if our data set is power enough to reconstruct mutation processes, at least in theory. For that, we simulated processes, and we varied two parameters. We varied spatial scale of a process, and we varied fraction, the variation of this process, a fraction of spatial variance that this process explains. And it turns out and so we kind of simulated these processes, then we sampled mutations from the simulated processes in a way that final set of mutations, like summary statistics, would exactly correspond to what we observed in top-met data set. And it turns out, at least in theory, if we look at processes at different scales and different fraction of explained variance, then we can confidently reconstruct processes that have scale of 10 kilobases and contribution of at least 0.3% to overall variability of mutation rate. So at least in theory, this approach seems to be quite promising for our top-met data set.
If you project known genomic covariates that influence background mutation rate into your new space, how well do they align to?
We'll discuss the question. But first, before we are going to go to there, that was not complete truth what I told about mutational process, because there is one additional feature that I didn't mention. It's strand-dependence of mutational process. So DNA is a symmetric molecule. However, some biochemical processes break this symmetry, and two of them turns out to be, for example, DNA replication and transcription. And in case of DNA replication, so DNA replication is a semi-conservative process, and two strands serve as a template for nascent strands. And turns out they're replicated by quite different replication machineries, including different DNA polymerases. And it's not surprising that these different machineries, they have different propensities to generate different types of mutations. So then, though two strands are kind of symmetric, when they're replicated, they have a symmetric accumulation of mutations. The other process is transcription, and let's say there is a transcription-coupled repair system. Vova mentioned it, and so this repair system, it senses transcribed regions, and it fixes damages on transcribed regions, but not on non-transcribed regions. That's why we have asymmetric accumulation of damages and asymmetric fixation of mutations. And so this, let's say, two processes, they're inherently asymmetric to strands. And so then we classify all mutational processes in those that are strand independent or that are strand dependent. Now I'll try to explain that we actually can devise a statistical procedure that can infer whether the process is strand dependent or strand independent from the data. One more important thing, just to go back, here the result of this procedure is not actually a mutational process yet. It is, let's call it, mutational component. It's not yet a process. It turns out if we consider complementary mutations separately, and let's say if we consider a process such as transcription coupled repair, then it depends on localization of a gene. And suppose that it tends to generate A to G mutation when gene is on reference strand. Then if we have a window and a gene is on reference strand, then it tends to preferentially generate A to G mutation. On the other hand, if a gene is on non-reference strand, then on reference strand, we tend to see T to C mutations. And the idea that we count only mutations on reference strand, then if a window corresponds to a gene on, if a gene resides on reference strand, then we expect this window has much more A to G mutation compared to T to C. So here each dot is a window, and we expect that that the windows that have gene on transcribed state, they kind of follow this way. They have many more A to G compared to T to C mutations. On the other hand, if we consider windows where gene resides on non-transcribed strand, then we expect that these windows have many more T to C mutations compared to A to G, and we expect that this window kind of lie along this ray. So, and the overall number of mutations reflect just overall activity of this process in this window. And from there, and this is pattern that we kind of theoretically would expect from a strand-dependent process. So basically, strand-dependent process generate not one component, not one dimensional component. It basically covers the whole space, the whole two-dimensional space, and it generates basically two rays. On the other hand, if a process is strand-independent, that it basically doesn't know anything about complementary mutation, and we expect that to have more or less similar number of complementary mutation in each window. And then if we look at rates of complementary mutations in each window, then they expect that they lie along the diagonal, and the overall number of mutations just reflect overall intensity of this process in a given window. And so this is, let's say, some theoretical thoughts about this, and there are two predictive properties that follow from this. One is that if a process is strand-independent, then we expect that it corresponds to a single component, and the single component has equal rates of reverse complementary mutations. Let's say in this case, we expect that it has equal rates on A to G and T to C mutations. On the other hand, if a process corresponds to, if a process is strand-dependent, then we expect that it corresponds to two mutational components, and they have identical rates of reverse complementary mutations between these two components. So this is examples what we theoretically,
So as you mentioned on the previous slide, we observed asymmetry in both transcribed strand, but also in terms of replicative convection. And in cancer, we don't really see any processes that have both classes of asymmetry. So you get 4 components when plotted here. For germline processes, do you see any that have both transcriptional and replicative asymmetry?
Yes, we also see in cancer, by the way, so this is a Vova can comment on that.
Yeah, so if you look on the, for example, on the processes that have very strong transcriptional asymmetry, like UV light induced mutation or smoking signature, it's half tiny bit, but very consistent rate of replicational asymmetry. And we believe that underlying biology of this is asymmetric resolution of the damage. So despite damage resides symmetrically on the ligand and ligand strand, during replication, conversion rate to mutation like 10% different. It's tiny bit different. But signal is very consistent across different type of lesions, and yeah, and it's always present.
Yeah, so what happens in cancer, because there are processes, Vova originally discovered apotheca symmetry with respect to replication direction. And there is Paul epsilon mutants, Paul delta mutants. Those show dramatic asymmetry with respect to replication direction. So the process we're talking about is quantitatively much smaller. So this is why nobody's focusing on that.
Yes.
Are these complementary, strand-dependent processes the main source of having, say, a two-dimensional eigenspace where you want to use ICA to find?
Yeah, exactly. This is the reason we use ICA to basically to align our independent components along these two rays. Yeah, this is underlying expression.
So for the discussion, we didn't think about it, but I was thinking maybe there is more elegant solution. You find some Jordan form or something else to do it. But yeah.
Theoretically, the basis recovered by NMF should also sort of form that convex hull of those two components, right?
Well, maybe a theory, but in practice it just doesn't work.
Might have just been a specific cost function you were using in NMF.
Well, we kind of. It seems that it's fundamentally does work. I tried this R package and 6-7 different versions of NMF with different kind of types of regularization. Seems they kind of give more or less the same. They're able to reconstruct, I don't know, 4, 5, 6 processes, but not 13 that we have here. Yeah, but so coming back to the strand dependency, this is actually a core statistical procedure here. So what does it mean that we have that process is strand independent? As I said, it means that it has identical rates of complementary mutations, and we can easily check it by looking at components. If a component is strand independent, that if we look at scatter plot of its mutation versus reverse complementary mutation, then we expect each point here is a mutation type. Then we expect that they should correlate. If they correlate, then it means that they're identical. On the other hand, if a process is strand dependent, then we expect that there would be two different components, so that rate of mutation type of one component would correspond to a rate of mutation type of complementary mutation type of another component. It might be not clear, but now it will be clear. So, and then we applied this, and when we just, for each pair of predicted components, we correlated spectra of one component and reverse complementary to spectra of another component. And what we immediately saw that basically all components were kind of stratified into classes. Either component is complementary, is correlated with a reverse complementary of itself, like let's say component 6 or component 6 or component 5. Or there were two pairs of components, such as one component is correlated with reverse complementary of another, but not with itself. So, and basically this picture first immediately classified components in pairs of components that correspond to one strand-dependent process and components that correspond to strand-independent process. And then we can also use these features for let's say, for different sorts of purposes, and one of them is to choose number of components, because we can run these algorithms with many different type of components, and for each we can predict different number of components, and then we can ask how many of them actually have this reflection property. How many of components have reflected component? And it turns out, as we increase number of extracted components, we first see that many of them pass reflection test up to this point, where if we extract 13 components, that all of them pass reflection test. This is the situation. And then we don't see any more, and then when we keep increasing number of extracted components, we don't see additional components that actually that have reflection property, meaning that probably this is the number of components that basically are biologically driven, and other components are just noise. So in contrast to this, if we look at our kind of traditional method to choose number of components from, let's say, eigenvalues or things like that, where basically this is number 13, it's not different from any other number basically here. So this is kind of powerful method that unambiguously selects number of components and unambiguously classifies components in strand-dependent or strand-independent processes. Yeah, are there any questions about this? To move on. Okay, move on. So it turns out that the predicted components are reproducible in a sense that they are robust with respect to bootstrap of windows. So we can do bootstrap of windows, predict components again, compare if they're similar to original one. Turns out, yeah, they're similar to original ones. Then we can run the same algorithms, but for different window sizes, for let's say window sizes of 10 kilobases or 100 kilobases, or for different, or even for different data set of population variation. And we also can see that predicted spectra of components, they are more or less similar. And then we can check if we see signal from these mutational components in de novo data. For that, we can ask if let's say regions where intensity of components are high show shift in spectra of de novo mutation that would mirror spectra of this component. And yeah, it turns out that it's true also for all these components. So indicate that they're robust. And then to your question, so now we have to understand what these components could be. And the first idea is just to correlate them with a bunch of genomic features or epigenomic features that reflect some biochemical properties of the genome. And there are a bunch of things that usually affect mutation properties, and among them are chromatin structure, DNA accessibility, timing of replication, or direction of replication, or gene bodies, direction of gene bodies, or different chemical modifications such as a methylcytosine or hydroxymethylcytosine. And this is basically a heat map of correlation of different processes with different genomic features, and we see kind of very distinct and diverse correlations. Let's say if we look at process, what we call process 1-2 is basically a process that corresponds to these two components. We can look at intensity of this process as just sum of intensities of these two components, or we can look at asymmetry of this process as like asymmetry, the difference of intensities of two components. And we can see that, let's say, for this process, we see a strong correlation with replication timing, DNA accessibility, and localization of gene bodies. But if we look at asymmetry of this component, that it's that it mirrors direction of gene transcription. And we can look at other components, and we basically see some distinct features. The same way they have different contributions to the overall variation of mutation rates, and they have dramatically different scales from around 10 kilobases to like almost 1 megabase. So then I'm just going to showcase some examples of mutational processes to make it clearer. So the process 1-2, which is strand-dependent process, and we think that this process reflects bulky damages. This is a spectrum of this process, dominated by enrichment of T2C mutations, and these are variability of two components that comprise this process along the genome. And we see that, let's say, two components, they kind of go in different directions, and they basically match gene directions. Here is a track of genes colored by their orientations with respect to reference strand, and we see that different components they peak at genes of a particular direction, and so these two tracks, basically the asymmetry of these two components reflect direction of a gene, and overall intensity of these components turns out to be similar to replication timing. And yeah, this is basically a part of this process is exactly a phenomenon that I described with transcription coupled repair. Well, transcription coupled repair is a part of phenomenon that is process 1 too. The other processes is actually asymmetric replication errors that we already just discussed. It is dominated by C2T mutations, and if we look at like variation of asymmetry of this process along the genome and compare it to predicted fork direction, actually fork direction is a quantity which reflect kind of to what extent the probability of being replicated as leading versus lagging strand of a given genomic regions. And we see that basically they're well corresponding. Correlation is high between direction of replication and asymmetry of this process. And the notable feature of actually direction of replication, it doesn't correlate with any other known biochemical property in the genome. So that's why this process has kind of unambiguous interpretation. It's likely really just asymmetric accumulation of mutations along the genome with respect to direction of replication. So then the other process reflect heterochromatin structure or replication timing. These two features turns out to be kind of statistically very similar. And so this process has quite degenerate spectrum. And the spectrum reflect that, with it just simply more or less reflect mutation rate, like almost uniform increase in all mutation types, with slight increase of C2A mutations. And again, if we look at intensity of this process along the genome, that it perfectly mirrors replication timing of genomic regions. So it doesn't suggest biological mechanism, but it suggests that at least we have biological association of this process, and it mirrors replication timing. So the other process has more interesting behavior along the genome. This is also strand-dependent process, and it shows like regional activity in large regions of the genome, and at the same time some notable spikes on genes. And it basically, first we noted that it is associated with fragile genes. It has a very specific spectrum. It's enriched by C2G mutations. But then we realized that this is also a specific process. And basically, it's not that we discovered this first. A few years ago, so the story was that, and Vova talked a little bit about this, if we quantify number of mutations that come from oocytes and from spermatocytes, it turns out that number of mutations from spermatocytes is around 3 times larger than number of mutations from eggs. So there are like 75 mutations that we observe that come from fathers and only 25 from mothers. But despite this, if we look at clustered mutations, there are very notable regions where mutations only come from mothers but not from fathers. And these regions were called maternal regions, and their signature was unique enrichment by clusters that come from mothers but not from fathers. In contrast to like genome, like overall expectations. And when we looked at maternal de novo clusters, it turned out that they perfectly corresponded to peaks of our processes. And when we quantified more formally, we indeed observed that if we arrange chromosomes by enrichment of what we call now maternal regions, we just decided to call this also maternal regions, then these regions are dramatically enriched by de novo clusters. So then we conclude that this process is associated with also specific mutagenesis. So we next looked at what happens inside these peaks inside genes, and it turns out that there are not only spikes of activity of this process inside genes, but also these spikes are asymmetric with respect to direction of transcription. Again, these colors show direction of gene transcription with respect to reference strand, and we see that two processes that comprise this also specific process, specifically peak at the peak specifically correspond with orientation of a gene, indicative that this process is asymmetric with transcription and likely associated with DNA damages. Then at the same time, it is also associated with asymmetry of replication strand, leading versus lagging. Then it turns out that this is a multi-scale process. If we, let's say, look at this process, then we can quantify its overall intensity, or we can quantify its asymmetry. And it turns out that scale of overall intensity is very large. It's like mega-base scale, but scale of asymmetry is very small. And indeed, we can look at it by eye. If we look at it, we see some small spikes that exactly correspond to gene and that reflect small scale asymmetric scale, and we see overall, and we see that spikes are positioned inside large regions where this process is active. Next we found that this transcription, this activity of this transcription process, this maternal process is actually inducing transcription-associated mutagenesis inside genes. And while transcription-associated mutagenesis had been described in Germline before, kind of the magnitude that we observed here people have never seen before. So it induces very strong transcriptional mutagenesis, transcription-associated mutagenesis. And finally, this, if we look inside these regions, that it turns out that they have dramatic acceleration of mutation accumulation with age, and specifically in mothers. So if we look at specifically null-size mothers, yeah.
Yes, so I just, is it okay to comment?
Oh, yeah.
So I think one piece of biology that people from outside biology may not appreciate here is that oocytes are fundamentally different from spermatocytes. So male germ cells, spermatocytes, keep dividing through life of a male. And oocytes are being arrested. And they are arrested in embryonic stage and they stay arrested non-dividing. So the age dependency of paternal age has been historically interpreted as number of cell divisions. I mean, we can talk about that, but there is evidence this is just number of cell divisions. So age dependency for maternal cells, for oocytes, is extremely interesting process because it's completely unrelated to replicating cells. So DNA damage, this is why this is a window into DNA damage processes.
Going back to this property, so if we just quantify increase of number of mutations in old versus young parents, that turns out that we see overall increase genome-wide, but increase in maternal regions is several times larger for maternal mutations. But if you look at paternal mutation, that we have more or less uniform increase in the genome in the maternal region. So this method allowed us to construct this high resolution track of intensity properties of this maternal process, and basically enabled us to make a number of insights that people couldn't do before, and paved the road to Vova's actual conclusion that large fraction of these mutations, this also specific mutations, they actually come from this two-step mechanism and activity of low fidelity polymerase. So then a couple of other processes. One we discovered is cytosine domination is C2T mutation in CpG context. It is well known and not surprising that we found it. But what is more interesting that we also found another sort of mutation in CpG context. It turns out that we found that there are C2G and C2A mutations in CpG context. And this is a process, as we showed, is a process of active demethylation of methylated cytosines. And basically, these two processes, they have polarized properties, while cytosine domination is active in methylated regions, and this is due to chemical property that methylated cytosine, they dominate at a much higher rate. On the other hand, the mutational process 10 is active only in non-methylated regions. And so from this, another test, we kind of thought about this as about this new process that it's active in low methylated regions and in regulatory regions because it actually because these regions they're low methylated because there is a process of active demethylation of these regions, and due to this active demethylation, we observe this mutagenic effect of this process, and we formulated basically we kind of supplemented a biochemical pathway showing that so following methylation of cytidine, this process of active demethylation, which has a byproduct of sometimes a generation of C2G mutations, these processes mutagenic to an extent that we observe this effect from unbiased analysis of our data set.
Couldn't it also be due to the fact that deaminating non-methyl C turns it to uracil, and something in the process of excising that uracil and replacing it incorporates the wrong complementary base?
Whoa,
Why is restricted to CPG sites? The question. So, non-methylated cytosines present everywhere, not in CPG contexts as well. And we see that this process is specifically active in CPG context. So it's reasonable to assume that it has something to do with CPG context. And actually we do a lot of tests comparing relation with CPG islands, for example, of C2A and C2G mutation rate in different contexts. And we found that acceleration is specific to CPG context. So it's reasonable to assume that it has something to do with this context, and most likely the reason what's going on, like the underlying mechanism of what's going on is demethylation.
I'd like to add to this because this is actually this is a great question. So because what we believe is happening, the downstream portion is exactly the same. So it's, I'm sorry for people with quantitative backgrounds for a little more biochemistry than is probably allowed at this meeting. So what's happening with uracil, right? So you deaminate cytosine, you create uracil. It's being repaired by BER, by basic excision repair. So glycosylase comes in, excises the base, creates a basic site, and a basic site is being repaired by basic excision repair. And if there is no access to the other strand, you create C2G or primarily C2G, that's REV1, for example. It depends on the repair pathway. So in this model, what's happening is that demethylation works in a very weird way, because you hydroxymethylate cytosine, and then you excise it completely, creating a basic site. So everything downstream from this thing is exactly repair of uracil. So this is why the type of mutation is exactly the same. So Vova's conclusion in this work that this is related to methylated cytosine is because this is very specific to CPG context and has very strong correlation with the rate of demethylation and hydroxymethylation.
That's super cool.
And biologically, what is cool here, it's a very localized process that doesn't occupy a lot of genomic space, but this is specific to regulatory regions. So people were asking about promoters and enhancers, and this is what drives mutations in this region specifically.
Yeah, and so we can describe a lot of more about biology about these processes, but I think enough biology for today. So, and again, we already discussed it. It's our matrix factorization strategy that basically show that we tried different things, and even more than we showed here. And it seems, for now, it was just optimal choice, and we could never squeeze more than 13 or 14 processes from the data. So yeah, that's basically it. And the short conclusions are that we observed 9 mutational processes. 5 of them are strand-independent, 4 of them are strand-dependent. For 6 of them, we have either biological interpretation or at least strong association. Yeah, it seems that all processes that we discovered are at least at scale of 10, 50 kb. But at the same time, even if we try to go down and to look for processes at 1 kb, we basically couldn't find anything. So we hypothesize that then even at small scales, likely there are no processes that actually contribute actively to mutagenesis. And that's basically it. And people who contributed, you you saw some of them today, basically. That's it.